And now, please welcome the chairs for our fellowship presentations, Nabeel El-Bardeesy and Sumera Ilyas.
Good afternoon, everyone, and we'll start with our fellowship presentation. So these are our 2025 Cholangiocarcinoma Foundation Research Fellowship awardees, and they will be presenting their progress reports or progress presentations. So first up, we have Ghada Nueria from Karolinska in Stockholm, Sweden, and she'll be presenting her project, which was focused on developing a blood-based test for diagnosis, prognosis, and monitoring of cholangiocarcinoma.
Hi, good afternoon. Thank you for presenting me. Let's start. So today, the diagnosis of cholangiocarcinoma relies basically on CA199 biomarker, which is non-specific, and MRI, which is which has low sensitivity at early stage of the cancer, but also has false negatives, especially on in the case of presence of chronic inflammation. One of these cases with inflammation is primary sclerosing cholangitis. This is a disease in the bile duct, again with chronic inflammation, which can lead to cholangiocarcinoma with a higher incidence of 600, up to 600 times more than the normal population. In the group where I am part of, in Karolinska, they are studying PSC, and they have done that for many, many years. So we have samples, yearly samples from PSC patients every year up to the development of the cholangiocarcinoma, which is a very valuable resource to study pre-cancer or early cancer. So we thought, okay, maybe we can have a simpler test, a blood test, where we can detect cholangiocarcinoma earlier. And our biomarker of choice was DNA methylation. This is a transformation that happens to the DNA. It's natural, but also it's a hallmark of cancer development and also the immune system response to the presence of cancer. So what we did is that we had samples from many centers in Sweden, but we also collaborated with the group, Trine's group in Oslo, Norway, and then we screened 1 million positions in the genome in DNA methylation, trying to compare them to non-cholangiocarcinoma, PSC, and to other healthy controls and to other types of controls to find few biomarkers that can be informative of the presence of cholangiocarcinoma. And as you see here, this was the discovery phase. So we started with only 95 samples. By comparing cancer to healthy donors and PSC, we found interesting results. And at this point exactly, I got the CCF fellowship, where I could actually validate my results in a larger cohort of 285 samples, which is this one. And this led to the development using machine learning and artificial intelligence, the development of two models, one here to distinguish cholangiocarcinoma from non-cholangiocarcinoma, so to diagnose, and another one to build a risk factor, a risk score, which can predict if these patients can be at high risk of cholangiocarcinoma, and then they have to be monitored and to come back for checks more often, or maybe they are fine and they can have longer times of without check. And here is one example to illustrate this. This is a gene in our genome, and the yellow line here corresponds to people with, healthy people, while the blue line corresponds to cholangiocarcinoma people. And you can see that there is a difference between them. So if we can screen that gene, then we can diagnose cholangiocarcinoma. In total, we had 800 such positions that are differential, and we could confirm 139 of them in the validation cohort. And the preliminary results, and I insist that they are preliminary, is that we can screen 66 probes, 66 DNA methylation positions to have almost a 100% efficacy in detecting cholangiocarcinoma. At the end, I would like to thank the Strati family for their generous donation for this work to be continued, and to also thank all my collaborators and all my funders.
Very nice job. We have time for 1 to 2 questions from the audience.
Yeah, I'll just ask a quick question. Are you able to go back into your longitudinal samples prior to cancer detection and sort of get a sense of how early, potentially, with these early results?
Yes, exactly. Unfortunately, the time didn't allow to show everything, but this is the point of the PSC up to longitudinal samples up to cholangiocarcinoma. So not only can we find the test, but also we can see how early can we detect. Is it just 1 year before, 6 months? And also it helps with the development of the risk score. Thank you.
And I know that the work is ongoing, but do you have any sense of how, in terms of what you've developed so far, how that compares to existing DNA methylation tests, or is that something you're planning to do?
Yeah, other DNA methylation tests, I didn't understand, sorry.
There are some that
In
have been
other
published.
cancers, you mean?
No.
In cholangiocarcinoma.
I can send you a paper.
Okay, good.
Great presentation.
Thank you.
Have you looked into iCCA versus perihilar cholangiocarcinoma in your methylation analysis?
Yeah. So that's also something that we are looking into. And with the new samples, we have more to compare, but we don't really reach the statistical significance with that. So we are aiming to team up with other people maybe and to do that. The second idea is to build a model that relies both on MRI and on the DNA methylation, so that we can at the same time detect and point out where exactly the cancer is.
Thanks.
Thank you.
Hello, beautiful presentation, congratulations. I know that some of your colleagues, like Gurolin in Oslo, actually are profiling the DNA in bile. Did you have the chance to compare the profiling in blood and bile in these patients? That might be interesting to see.
Yes, it is interesting. We didn't do that yet, but also we keep in mind that we are basically looking into immune cells here, and not really something that is immediately coming from the cancer. Thank you.
Okay, we'd like to thank Nuaria.
Thank you.
Okay, the next talk is by Donggi Moon from Mayo Clinic in Minnesota. And his talk is also on early detection. Here, another modality, novel glycopeptide biomarkers.
Yeah, thank you for the introduction. And thank you for giving me opportunity to share our result on developing glycopeptide biomarkers for diagnosis of cholangiocarcinoma. So as most of proteins that we heard and we learned yesterday and today, and proteins that we are studying are cell surface protein, and those are, which means those are glycosylated proteins, and glycosylation is one of the most abundant PTMs, and is known, aberrant glycosylation is known to associate with cancer. And so, and as you can see, I study one specific type of glycosylation, which is N-linked glycosylation, where sugar or carbohydrates attached to one amino acid, which is asparagine, depicted as N in this picture. And this shows some, and following some rules, they can be attached in protein. And just for today, just remember this pinkish diamond and red triangle, which are sialic acid and fucose. And those are known to be associated with cancer. And already we are using carbohydrate or sugar or glycoproteins as a biomarkers using blood test. And as an example, CA99 again contains the sialic acid and fucose are being used for diagnosis of cholangiocarcinoma or pancreatic cancer. And some subfraction of AFP, which is AFP-R3, is also fucose-related glycoproteins and used for diagnosis of hepatocellular carcinoma. And C8-125 or MOX16 is a different type of glycosylation, which is called O-glycosylation, is also being used for diagnosis of ovarian cancer. So, and as I just told, so CA99 is being used for diagnosis of cholangiocarcinoma, but which has some limited in sensitivity and also specificity. And most importantly, some of the human population does not secrete CA99, which is Lewis antigen negative, which can be resulted in false negative in diagnosis. So that's the reason we are trying to develop some another potential novel glycoproteins that can be used for diagnosis of cholangiocarcinoma. For doing that, we first do some discovery study to find some target glycopeptides. And then we developed some mass spectrometry-based method to measure those glycopeptides with the aberrant expression in serum samples, and we validate in some large cohort samples. So this is a discovery study that we did using tissue and serum samples, and we extract protein from those samples and making peptides and enrich glycopeptides using some chemical method, and measure those glycopeptides in mass spec, which means we can identify and quantify those glycopeptides in those samples. And among those, in tissue study, we identify quite a good number of glycopeptides, up to 5,000 glycopeptides in those samples. And we can, as you can see in this plot, this green is normal and red is cholangiocarcinoma tissue samples, and we can see some quite clear separation among those. And among those, we identify some 800 glycopeptides that actually overexpressed in tumor samples. And again, those are quite good number of proteins, around 80% are fucosylated and sialylated glycosylation, which again known to be associated with cancer. And in serum study, we also pick good number of glycopeptides that actually shows quite high overexpression in cancer patient. And particularly, the most important thing is in this study design, we include this non-secretory group, which means does not secrete CA99, and those glycopeptides actually secrete in those patients as well. So after selecting some good target, 4 target of glycopeptides in our discovery study, we did a targeted study using, again, mass spec. And this is the summary of the result. So each row represents one glycopeptide. So we targeted 4 glycopeptides, and each column is 1 individual. So in cholangiocarcinoma patient, we detected in most of those glycopeptides, sometimes 1 or sometimes 4, but it can be used in complementary. And most importantly, we can detect those glycopeptides in population of CA99 negative group as well. And control sample, there's small, false negative cases, but most of them, we did not detect these glycopeptides. And basically, we can achieve like 80% of sensitivity and 90% of specificity. And in conclusion is, we developed a mass spec-based method for discovery and also targeted method. And then further study are required to correlate the clinical data and along with some kind of complementary use of these markers with CA99 for sure, and also we learn from DNA methylation as well, so it can be complementary use. And thank you for my 2 mentors, Dr. Akhilesh Pandey in the mass spec side, and also Rory Smith for clinical side, and other team members in Mayo Clinic. And most importantly, thank you for Cholangiocarcinoma Foundation to give me opportunity to do this basic science as I'm a chemist and mass spectrometrist, and to give me opportunity to contribute to cholangiocarcinoma field. Thank you.
So do we have questions?
Hello, thank you for your talk. So I'm wondering if you evaluate the level of glycosylation as well in each protein, and if so, how the high glycosylation affect the function of protein?
Yeah, I got some similar question many times, but and a very good question, a great question, I think. So you're trying to correlate with some clinical data first, some tumor stage, or with some level of these glycosylations. And so far, I could not see any clear correlation on these 4 glycopeptides, but mainly because I targeted only those 4. But we have like 800 or so glycopeptides that are known to, that are overexpressed in this patient. But I think further study are required, some kind of mechanical study. What is the reason of this aberrant overexpression on those samples? But yeah, thank you.
I just have a question. Did you do a comparison with non-cancer liver injury states? I didn't notice on your slides.
Yeah.
You did, okay.
Yeah, so you mean the non-cancer liver disease patient. Yeah. We did, but I didn't include in this slide,
Yes.
but we also have some subgroups with liver disease patient.
And right, and this is the negative
Yeah.
control, yes.
Yeah.
Okay.
Thank you.
Thanks for the great talk.
Right, next we have Anahoa Lapitz from San Sebastian, who will be presenting her fellowship project on the development of a polygenic risk score to predict recurrence and survival in patients with resected cholangiocarcinoma.
Okay, so good afternoon again. Now this time I would like to thank the Cholangiocarcinoma Fellowship for giving me the opportunity to conduct this project. So currently, the only potentially curative treatment for cholangiocarcinoma is tumor resection and/or liver transplantation. However, despite curative intent surgery and the use of adjuvant chemotherapy, recurrence rates remain very high, with around 50% of patients developing a recurrence episode from 10 to 20 months after surgery. Therefore, there is an urgent need to find some prognostic biomarkers that would allow us to predict recurrence and overall survival after surgical tumor resection in order to assist clinical decisions. Recent studies have published so many prognostic biomarkers for cholangiocarcinoma. However, these studies can have many limitations. Most of them, they are single-center studies, include small patient cohorts, and rely on semi-quantitative techniques, such as immunohistochemistry, resulting in a limited statistical power, a bad representation, and a potential observer bias. Therefore, in this study, we have done a multi-step procedure combining literature review, meta-analysis, and an international multicentric large cohort validation by quantitative open array technique. Validated biomarkers were then integrated with clinical pathological variables by machine learning in order to develop some predictive models to predict recurrence and overall survival after surgery. These predicted models were then incorporated in a web-based clinical calculator in order to obtain personalized risk prediction. From the literature review, we observed or we identified 496 genes and proteins associated with cholangiocarcinoma prognosis. We evaluated the association of these candidates with overall survival across 5 independent transcriptomic cohorts and select 45 genes associated with a bad prognosis and 7 with a good prognosis. We then validate this candidate in a large multicentric international cohort of around 200 samples, and we were able to validate some genes for recurrence and overall survival, which were independent on the resection margin status, the lymph node invasion, the tumor stage, the cholangiocarcinoma subtype, and the sample of origin. From this analysis, 9 genes were commonly associated to both outcomes, recurrence and overall survival. We then combined these biomarkers by machine learning models, and we observed that the random survival forest gave us the best results. We then combine different 4-gene combinations and perform a final model of only 3 genes and 4 clinical variables that was able to predict recurrence and overall survival. Indeed, this predictive model predicts recurrence and overall survival with a high accuracy, as we observe a high correlation between the predicted and the observed outcome. Interestingly, this model allowed us to stratify patients among 3 main risk groups: a high-risk group with early recurrence and a poor overall survival, a moderate risk group with median outcomes, and a low-risk group with a low risk of recurrence and a median overall survival exceeding 10 years. In order to try to get closer to a potential clinical application, we then developed a web-based calculators, where clinicians can input clinical variables and gene expression data obtained by RT-qPCR, and they will get an individualized risk prediction of each patient for recurrence and overall survival, helping the patient management. In conclusion, we have identified and validated a prognostic tissue biomarker panel for resected cholangiocarcinoma. The integration of these models with clinical variables allowed us to accurately predict recurrence-free survival and overall survival. Then were integrated in a web-based calculator that helped individualize predictions of postoperative recurrence and survival. And currently, we are evaluating if these models can predict the benefit from adjuvant chemotherapy, supporting personalized postoperative decision-making and future clinical implementation. Finally, I would like to thank our research group in San Sebastian, all the collaborators that are patients. So thank you very much for your attention.
Well, thanks for the excellent talk. Is there questions from the audience? I have a question if nobody has. I noticed, I think 2 of the 3 genes were the glucose transporter and lactate dehydrogenase. What was the third?
Yeah, the third one was a fasting one, which is a linker from which links the actin filaments. Okay. So it's involved in motility and invasion.
Right. So I was going to ask regarding the other 2 whether if there's already a known correlation between FDG PET before resection and for prognosis, because this looks like presumably a glycolytic signature, at least from these 2 genes. So just curious.
Yeah, so thank you very much for that comment. It's really interesting. And I think that it has something related to that, because the first gene was lactato dehydrogenase 1, which is the gene that encodes an enzyme that transforms pyruvate to lactate. So it's related to anaerobic glycolysis. So therefore, the glycolytic pathway may for sure play a role in those patients with a worse prognosis or a more the differentiated tumors. Yeah.
Yes, and then you also had the glucose transporter, which would go hand in hand. Yes.
Both are related, yeah.
Okay, well, thank you again.
Thank you.
So the next talk is by Soravis Ostafan from Beth Israel in Boston, and he's going to speak about spatial transcriptomic analysis of the tumor microenvironment.
All right, thank you, Dr. El-Bardeesy, for that introduction. So today I'll tell you about distinct tumor microenvironment in cholangiocarcinoma with different anatomical subtypes. So as you have heard, different cholangio subtypes are very different, and where intrahepatic and distal can have very different genomics makeup, and even dicellular makeups are also different. But the key questions that we do not understand is, are there differences in tissue cellular organization across these different subtypes? And the significance of this knowledge is really understanding immune organization can help us understand who would respond to immunotherapy and also which immunotherapy we may want to use. So this is the study outline. I've identified resected tissue from intrahepatic perihilar and distal cholangiocarcinoma, and there's now this technology called spatial profiling device where cells can be measured within its organization and measured at single cell level, and we can look at that gene's expression. And my study utilized a single genomic platform called G4X, which is a measured transcriptomics of 500 genes in which I have customized to be specific to cholangiocarcinoma tumor microenvironment and the cell that we would like to investigate, and also get a fluorescent H&E output. And it's a really scalable platform where you get up to 128 samples in one run. And suffice to say, we got high quality data out of this, and we got up to 2 million cells from profiling 46 samples. And so how do you go about understanding this? So this is an example of a UMAP, and each of these represent a dot. And what you're trying to understand is if you imagine a dot is a face, and each face has 500 features, and you ask people to separate around the room where people who look alike are closer to each other, then you get formation of these groups. And these groups are really driven by cell types. So for example, here we have hepatocytes that likes to express albumin. And you can see that all of them are in this one corner. And with this technique, we're able to label cholangiocyte, cholangiocarcinoma, endothelial cell, macrophage, dendritic cell, and neutrophils. We are also able to interrogate subtypes of cancer-associated fibroblasts because of our panel, including matrix-forming, antigen-presenting myofibroblasts, and also inflammatory fibroblasts. We are also able to look into the different cell types, including T cell, B cell, plasma cells. And after labeling all of these cells, we then ask, well, are there any difference in the cell numbers within the different anatomical subtypes? And we found that distal cholangiocarcinoma have more B cell and plasma cell, whereas intrahepatic subtypes has more myofibroblasts. However, these are relatively known information because we have a lot of data from single cell. What this technology really does is you can in situ project these cells back into its native location. So for example, this is a normal liver section in which we're able to profile 280,000 cells. And you can see these are mainly hepatocytes that are well organized. And you can see that the bile duct within the liver are small and not as well organized. And you also have these nice endothelial lumen here that are forming. And you compare this to a common bile duct, which is where distal cholangiocarcinoma arises. And you can see that the structure of cholangiocyte organization is very different, where these blue dots are representing cholangiocyte organized in the distal bile duct. And now you have the surrounding fibroblasts that are forming. And now if we look into cholangiocarcinoma, for example, in this intrahepatic samples, you can see that in the bile duct that has cancer, the ducts are forming, and then it's really have reactive cholangiocytes around it. There's T cells that are infiltrating, and there's myofibroblasts here, and the liver is sort of pushed to the other side. And if you look at distal cholangiocarcinoma, you can see that the ducts are much bigger and is surrounded by myofibroblasts. And so now, how do you get to understanding these 2 million cells? So you can ask an AI model using a neural network to understand, are there recurring patterns of spatial organization? And this is essentially a cell niche analysis where you are asking which cells likes to be close together and kind of like are friends with each other. And what we found is tumors have two distinct friend groups. One type of tumor really likes to be on its own and doesn't have immune cells around it, versus there are tumors where there's a lot of immune cells. And if you project this back into the tumor, you can really see that, so on the left here, this is a regular cell annotation, but here's the niche, and you can see that in these samples, there's a strong niche, but there's a part of the tumor where there's no contact with the immune cells, indicating that it could be resistant to immunotherapy. You can do the same for large duct, and here in the large duct, there's a lot of immune infiltration, particularly with B cells. And you can ask which organization is unique in each subtypes, and you really see that immune exclusion is more common in intrahepatic. And also, we are able to capture tertiary lymphoid structure using this technology. And these are mini lymph nodes that helps kill cancer. And so in summary, we have generated a high-resolution spatial profiling and shows an architectural organization of cholangiocarcinoma, and have detailed the findings of each subtypes. And now we are working on understanding are these findings true in different parts of the tumor from the same patient, and also can they predict response to immunotherapy. And just to thank the spatial technology unit at the Broad and BIDMC, my clinical team, and also our collaborators who we are working on acquiring more tissue, including patients treated with durvalumab, and also patients from Thailand who has liver-associated cholangiocarcinoma, liver full-associated. All right, thank you. And I'm grateful for the funding from Cholangiocarcinoma Foundation, and I was funded by AstraZeneca.
That was beautifully presented and explained, so congratulations. I'm interested in the antigen-presenting fibroblasts, and some investigators use ApoE as a marker. I couldn't tell which marker you're using.
Right. I don't know if I can go back quick enough for this, but it's mainly HLA-DR that's forming. But really, Dr. Gore, what's coming up here is that it forms its own UMAP cluster. That is really the most unique part of the antigen presentation that I decided to label them as antigen presentation. Because if you, all right, we're back here. But if you look at this, the antigen presenting fibroblasts are like right between where most of the fibroblasts is and where the macrophage is. And because they're right in this transition, I think it's right to label them as antigen presenting. And it's HLA-DR here, but obviously we have information on all the 100 genes that you can then look.
And second, do you see the antigen-presenting fibroblasts in normal liver and normal bile ducts?
Great question, and I think there are some that got labeled, as you can see here. Yeah, there's some pink clusters even in the normal tissue.
But they're more prevalent in the cancers, I guess.
Correct. Yeah.
Sravis, really nice job. So some of the populations that you've identified, like the MyCAF population, have you gotten to the point of, I mean, you've done niche analysis and looking at what's in their neighborhood, but the populations that you've highlighted for the different subtypes, do you know what are the populations that they're really interacting with? For instance, the MyCAF and what signaling pathways those are leveraging?
So that's our next step is now to bring in like ligand receptor interaction, which can then also tell us exactly which gene is interacting with what cells.
And the plasma cells, are they like actual plasma cells or are they plasmablasts? Is it an immature phenotype?
I don't want to tell you the wrong things, but we can definitely can look into that. But it's clear that there's tertiary lymphoid structure that drives those plasma cell labeling. Yeah.
Okay, excellent. Thank you so much. Next, we have Binbin Lee from Mayo Clinic in Rochester, Minnesota, who will be presenting his work on chemotherapy-induced senescence promotes tumor immunosuppression in cholangiocarcinoma.
Hi, good afternoon, everyone. I'm Binbin from Mayo Clinic, and today I'm going to be talking about the negative effect of chemotherapy and the key effector of DDI-15. Cholangiocarcinoma is highly lethal, and even with current standard of care, the outcome remains very poor, suggesting that tumors rapidly adapt to the treatment. So we focused on one possible adaptation, senescence. Although senescent cells, they stop dividing, they are metabolically active and secrete set of factors that reshapes the tumor microenvironment. So we hypothesize that DMCs induce tumor senescence, which drive the immunosuppression and the disease progression. So first, we want to know what cell becomes senescent. Using P16 or P21 staining, we found that the senescent cells are predominantly tumor cells rather than the stromal cells. And importantly, the chemotherapy increased those senescent population, suggesting that the treatment is not only killing tumors, they're also enriching senescent tumor population. Next, we want to test whether these senescent tumor cells are functionally important. So in mouse models, we selectively depleted either P16 or P21 positive senescent tumor cells. In both settings, the tumor burden were reduced. And meanwhile, the suppressive tumor macrophages were reduced, while cytotoxic T cells increased, suggesting that tumor cell senescence actively drive immunosuppressive microenvironment. This is also clinically relevant in two independent cohorts. Higher senescence signatures are both associated with worse overall survival. Consistently, so in mice, removing these senescent tumor cells significantly enhanced the chemotherapy efficacy, suggesting that tumor cell senescence contribute to the treatment resistance. Next, we looked for the set of factors driving this effect. Using a protein array, we identified the GDF15 as a predominant candidate, supported by the single nuclear RNA sequencing from patient samples. And functionally, in mice, higher GDF15 is associated with the larger tumor, increased suppressive macrophages, and decreased CD8 T cells, suggesting that GDF15 bridges the tumor senescence with immunosuppression. In summary, chemotherapy induced senescence in CCA, and these senescent cells secrete sensitive factors, especially GDF15, that promote the suppressive macrophages and weaken anti-tumor immunity. Targeting senescence or GDF15 may therefore improve the treatment response in CCA. And finally, I would like to thank the Merck CCF Fellowship for support, and thank you for your attention. And I would be very happy to discuss this further at my poster. Thank you.
Professor Gretton.
Thank you. That's very nice. One of the problems with targeting myeloid cells is that they come back right away. So I was wondering, have you done, if you treat these mice and deplete the factor, do you know what's going to happen to the macrophages? Are they going to repopulate in the tumor afterwards?
You mean depleting the macrophages?
No, you're targeting the senescent cells. So if you take away the senescent cells?
Yeah, if we take away the senescent cells, we saw decreased macrophage.
And is this going to last? Is the effect going to last? That is what I was wondering.
Yes. Yeah, we have both in vivo and in vitro data showing that. Yeah, especially the M2 macrophages.
Thank you.
Yeah.
So I have a question. In your model, you suggest that the impact is mediated through, it's me. Yes. That it's mediated through immunomodulation. Have you done the same experiment by depleting the senescent cells in immune deficient settings to formally show that there's an important component to the immune modulation?
That's a good suggestion. Yeah, so we will consider that. We haven't done that. Yeah.
Back here. Really great talk. I was wondering if your data showing that chemotherapy-induced senescence really upregulates GDF15 has implications for timing of therapy if you're to think about translational studies. So have you explored like different sequencing? Would you give a GDF15 blocker concurrently with chemo? Would you have to pre-treat with chemo in order for it to come up to get the maximal effect? I'm wondering if like that's something that needs teased out on a more systematic level before you would propose a trial as a result or something.
Yeah, that's a good suggestion. So yeah, we will consider doing that. Yeah.
Well, so in terms of sequential, I think the GDF15, the reason I don't think that may be the best route for targeting it because that's one factor in the SASP, right? So if you deplete that, it's possible that there's another SASP factor that emerges. So what we're trying to do is actually target those P16, P21 positive cells. So we're using an LNP approach to selectively target those cells themselves.
Hey there, great presentation. Thank you so much. Noting that in the clinic, we traditionally stop GemCis after an induction period of 6 months in the first-line setting. My question is, what is the durability or the reversibility of these senescent changes when you remove the selection pressure of chemo, like we do clinically?
So senescent cells, they will not come back, but they are there. They will not die. They're secreting bad factors affecting the tumor microenvironment. So it's ideal to clear those senescent cells.
Okay, thank you.
Thank you.
Next, we have Justin Lowe from Vanderbilt, targeting PIN1.
Hello. Thank you so much for the opportunity to speak. Excited to be sharing my work. So to start, PIN1 is a peptidyl-prolyl isomerase enzyme. It's overexpressed in various tumor cells and cancer-associated fibroblasts, and that leads to activation of oncogenes, as well as contributing to a immunosuppressive tumor microenvironment. There are existing methods for targeting PIN1, but some of them, like all-trans retinoic acid, are not molecularly specific, and small molecules like sulfapin are not intrinsically tumor-specific. And so we became interested in targeting PIN1 using siRNA therapeutics, which are molecularly specific and can be engineered to be tumor-specific as well. Specifically, we employed siRNA lipid conjugates, termed siRNA L2, which we have previously optimized to reversibly bind to endogenous albumin protein in the blood, as a way of extending the circulation half-life and taking advantage of aberrant uptake of albumin into tumors. The structure and the additional stabilizing modifications are shown at the top right, and we have further shown that there's robust uptake and delivery of the siRNA L2 in various models of cholangiocarcinoma. Two of them are shown here, the KRAS P19 mouse model and the HUCCT1 xenografts, where the siRNA shows up as red. Next, we performed an siRNA screen to identify a lead sequence that targets PIN1, starting with a broad screen in tumor cells with regular siRNA. We varied the location and length of the sequences. We moved forward with the 4 sequences that led to the most knockdown of PIN1, and confirmed that these also mediate silencing of PIN1 in cancer-associated fibroblasts derived from the KRAS P19 model. And then finally, we synthesized these as siRNA L2 conjugates and showed that without any transfection reagents, we're still able to get knockdown. And from this, you can see that the PIN1 286 sequence performed the best in all of these assays, and so we moved forward with that. Next, we looked at the tolerability of this in healthy mice. And so we treated them at 20 mg per kg with the PIN1 siRNA L2 compared to saline, and then we also threw in a couple other of the methods of targeting PIN1 as well. With the PIN1 group, we did not see any changes in body weight, nor any significant alterations in blood counts, in plasma tests, nor on histology on H&E of healthy organs. And here, this therapeutic testing is obviously very much a work in progress, but I performed a pilot experiment in the KRAS P19 hydrodynamic tail vein injection model of intrahepatic cholangiocarcinoma, treating them with 10 mg per kg, PIN1 286, siRNA L2 weekly, or saline. Measured tumor burden in 3 different ways using a bioluminescent reporter, as shown here on that graph in the representative images, as well as by the liver body weight ratio and percent tumor area in the liver cross-section. And these were congruent in showing slowed tumor growth with treatment. Since the goal is to knock down PIN1, I also graded the PIN1 IHC staining. You can see the representative tumors there and the lower H score for PIN1 expression. And then finally quantified the T lymphocytes and macrophages, showing a trend towards greater representation of these immune cells in the treated group. Finally, we also wanted to better understand the landscape of PIN1 expression in human cholangiocarcinoma, so through a large collaborative effort. This is a tumor microarray from Vanderbilt cases, and all subtypes of cholangiocarcinoma are represented here. On IHC, the PIN1 showed at least weak and ranging to very strong expression of PIN1 compared to normal tissues. You can see the different patterns of tumor cell and stromal cell expression of PIN1 here. And then a couple nuances to highlight is that there was some spatial variation between 2 different cores from the same specimen, as well as a temporal variation where patients were represented more than once, and in these cases often had more PIN1 when the tumor recurred. So to summarize, we designed a PIN1 targeted siRNA L2 conjugate. It silences PIN1 in vitro. It's well tolerated in healthy mice, distributes into cholangiocarcinoma tumor models, and preliminarily may have a therapeutic effect. In addition to scaling up those studies, I'm actively exploring whether this can be combined with chemo and immunotherapy, and of course, taking a deeper dive into knockdown by cell type, by single cell analysis, and immune cell characterization. With that, I'd just like to thank the CCF Community Funded Research Fellowship and George Riddle for their support of my work, as well as all the lab members, mentors, collaborators, core facilities, and patients who contributed. Thanks so much.
Thank you. Do we have any questions? Let's just take one question just because we're behind in time. Please go ahead.
Good work, Justin. Very interesting. I was wondering if you have a way to look at the cell specificity of your silencing RNA, or is because PIN1 is mostly in the tumor, you don't really care between the tumor and the non-cancerous tissue?
I think fortunately there's not much PIN1 expression in the normal hepatocytes, for example, anyway. But I was lamenting this at my poster. I just don't have the images, but we do have methods for actually quantifying the siRNA accumulation in different organs. I think for the most part it's very low. We used what's called a peptide nucleic acid assay, which could quantify on the HPLC exactly how much is in there. And so the tumor accumulation is good. The hepatocytes really do not take up very much. I believe there is some amount that is not specifically taken up by Kupffer cells, but I'm trying to separate out that a little bit more. But that's most of the non-specific liver accumulation, and other organs like lung and kidney extremely low.
Thank you very much, Justin. Next we have Farnaz Bargi from MD Anderson presenting her project on targeting KRAS in biliary tract cancers.
Yes, good afternoon, everyone. I am Farnaz Bargi, a postdoctoral fellow at MD Anderson in Dr. Merck Benstam's lab. Today I'm sharing our findings on therapeutic targeting of KRAS in biliary tract cancer, focusing on cholangiocarcinoma. Yes, we are interested in targeting KRAS as well. I am referring to both Dr. Azad's and Dr. El-Bardeesy's talk. Just briefly going through the history of KRAS, targeting KRAS for a long time. The molecule was undruggable, with the discovery of switch 2 pocket on KRAS, it was a major breakthrough that led to the development of FDA-approved KRAS G12C inhibitors in non-small cell lung cancer, where patients have benefited the most, but the variant G12C is relatively low in other types of cancers, mainly in BTC and pancreatic cancer. And currently, KRAS is being targeted broadly beyond G12C in clinical trials using RAS(ON) inhibitors, both the pan-RAS and G12D selective drugs. It is exciting that currently the pan-RAS inhibitor in the clinic has doubled the overall survival in PDAC patients. So this is really exciting for us to have the KRAS inhibitors enrolled in clinic for BTC cancer as well. So in BTC, KRAS alterations vary by subtypes, with the highest frequency in extrahepatic cholangiocarcinoma, and they are dominated by both G12D and G12V variants in BTC. So that is where our first objective comes in. In this project, we wanted to target KRAS beyond G12C using the 2 sets of compound in our cholangiocarcinoma models. Moving on, in vitro studies, we showed that the pan-RAS inhibitor and also G12D selective inhibitor, they are doing selectively and more active in KRAS mutant cell lines and in G12D mutant cell lines, respectively. And we confirmed this selectivity by generation G12D isogenic BT cells and compare the IC50 of the isogenic cell to its parental cells. And we also we did some proliferation assay, and we confirmed that the pan-RAS inhibitor is more active in KRAS mutant CCA cells. The same thing for the G12D inhibitor. Now we significantly showed that the drug is more active selectively in EGI1 model, which is mutant for G12D. So moving on in vivo studies, we generated a CDX model, cell-derived xenograft, derived from EGI1 cell line. And we showed that the pan-RAS inhibitor induced partial responses initially. However, towards the end of the daily treatment window, tumors grew back. However, the drug prolonged survival. And moving to other model, this time we used the PDX model carrying G12V mutation, and we saw that the pan-RAS inhibitor induced partial responses and prolonged survival in this model. However, using a different model, the PDX model, with G12D mutation, the drug didn't induce tumor, didn't induce tumor regression, So altogether, we conclude that the RAS(ON) inhibitors are active in CCA models in vitro. However, monotherapy shows limited activity in some CCA PDX models. So this highlights the need for combination therapies. So this is where our second objective comes in. So referring to Dr. Merrick Bernstam talked today about the ADCs and their potent activity across multiple tumors, and also since TROP2 is already expressed in BTC, so we hypothesize that the combination of these two drugs, RAS(ON) inhibitors and TROP2 ADCs, may enhance therapeutic efficacy in CCA. And the future directions include combination studies, which are in progress, and also we are doing both RNA-seq and proteomics analysis to identify adaptive responses and potential combination targets. At the end, I am thankful to my lab, all colleagues and collaborators, and in particular my mentor, Dr. Merrick Bernstam. Last but not least, I am deeply grateful to Team Cure Cholangio for their support and dedication to make the fellowship award happen to me in honor of Arturo Mendoza, Penny Ziegler, and TJ Wallace. Thank you.
Any questions? Oh.
Great presentation. I only have 2 comments. Completely agree on the combination therapies. With the PDX and CDX models, just one caveat. I mean, there may be a role for the immune system. I mean, there's some papers that have shown that. So I wouldn't say that, you can't conclude that it doesn't work as well because that factor is out,
Okay, thank you for your comment. But we are using multiple PDXs, so I'm just showing two of them. That one of them doesn't work, one works. But I appreciate your comment.
So thanks for the great talk. We'll move on to get back on time. Thank you. So next it's Georgios Koulios, also from Vanderbilt, and he's going to talk on inhibition of tropomyosin receptor kinase.
Thanks so much for the kind introduction. Once again, I'm Georgios Koulios. I'm from Greece, currently research fellow in Youngmin Lee's lab in the Department of Surgery at Vanderbilt University Medical Center. My mentor's lab has recently identified a druggable growth signaling pathway where hepatic stellate cells release neurotrophin-3, which is a neuropeptide and a member of the nerve growth factor family that induces hepatocyte proliferation through paracrine tropomyosin receptor kinase B signaling. In the brain, TRAC receptors are really well described and are divided in 3 high affinity types, TRAC A, B, and C. And what is special about neurotrophin-3 is, although it shows highest affinity to TRAC-C. It can bind to all 3 TRAC receptors. On the right-hand side, we identified we also identified that stellate cells release another neuropeptide called brain-derived neurotrophic factor, short BDNF, that shows highest affinity to TRAC-B and has similar effects as NTF3. In our working model, we hypothesized that hepatic stellate cells give rise to cancer-associated fibroblasts that release BDNF and NTF3, which induce fibrosis in the tumor microenvironment and proliferation of cancer cells through autocrine and paracrine drug signaling, respectively. And we believe that the administration of FDA-approved TRAC inhibitors, such as larotrectinib with the brand name Vitrakvi, could disrupt that proliferative effect towards both the calves and the cancer cells. Gene expression analysis from the TCGA dataset showed overexpression of NTRK1 and NTRK2, which are the genes encoding TRK-A and B, in cholangiocarcinoma tumor samples, as well as overexpression of BDNF. In an independent cohort of 122 patients, we observed overexpression of the NTRKs as well as their ligands, including NGF, which is another neurotrophin, as well as NGFR, which is a gene encoding the low affinity TRK receptor, an NGF receptor. And notably, this overexpression was in about 1/3 of those patients, and also belonged in the immune classical and inflammatory stroma subtypes that are interestingly also more fibrotic than the other 3 cholangiocarcinoma subtypes. On the protein level, we observed overexpression of TRAC-B, TRAC-C, and NTF3 in tumorigenic lysates compared to the non-tumorigenic ones, as we can see, for example, in patient 2 and 4. And then in human cholangiocarcinoma tissue, by using reelin as a hepatic stellate cell marker, we could show expression of BDNF and NTF3 by immunofluorescence, which was further supported by immunohistochemistry, where we saw expression of the TRK receptors as well as their ligands. We have been able to generate patient-derived organoids from cholangiocarcinoma surgeries, and treatment of those organoids with NTF3 significantly increased proliferation compared to the vehicle-only organoids, an effect that was significantly decreased by co-incubation with larotrectinib. And comparable results were observed using BDNF instead of NTF3, suggesting that NTF3 and BDNF induce proliferation in a TRK-dependent manner. I would like to thank my mentor, Youngmin Lee, as well as my colleagues, Ting-Fang Li and Kevin Ray, for their support and guidance throughout our research project. And last but not least, Cholangiocarcinoma Foundation for making that research project possible through the research fellowship. Thank you so much for your time.
Georgios, very nice talk. One of your earlier slides, you had mentioned that the CAF and TRK signaling has immune modulation effects. So which effects have you seen? What cell types is it acting on?
We haven't done studies so far regarding immune reactions, but is that what we know so far regarding CAFs, that one of the functions for inducing proliferation and fibrosis in the tumor environment is also immunogenic effects, but we haven't done yet studies on that.
And do you know which CAF subtype?
We also don't really know. It could be a pretty interesting next step to do studies and testing the different types of cancer-associated fibroblasts that are active in that interaction.
Any other questions? All right, thank you.
Thanks so
That was
much.
very nice work. All right, next we have Davide Pavro from Mayo Clinic in Rochester, who will be presenting his project on BAP1. Loss of function protects mitochondria and confirms cholangiocarcinoma resistance to ferroptosis.
Good afternoon, everyone, and thank you for the opportunity to present our progress today. So we know that cancer therapy aims to essentially stop cancer growth by inducing cancer cell death. And over the past years, several different cell death modalities have been tested, but unfortunately, the efficacy has not been or has been often limited. And so here we focus on ferroptosis, which is a metabolic pathway to cell death and intersects with different cancer hallmarks, including metabolism, oxidative stress, and immune evasion. So ferroptosis is this iron-catalyzed form of cell that is driven by phospholipid peroxidation, which eventually disrupts and cause rupture of cellular membranes, inducing lytic cell death. And so over the past few years, we tested several murine and human CCA cell lines, and the majority of them are sensitive to ferroptosis inducers that we tested. We also tested two specific ferroptosis inducers in our preclinical liver orthotopic CCA models, and they were also capable of reducing tumor burden and induce lipid peroxidation inside the tumor, which is what we want to see. And lastly, a recent study in a patient-derived xenograft, a combination with a ferroptosis inducer and standard of care GemCis, significantly reduced tumor growth in a PDX that was resistant to GemCis. So we also have a platform, as a collaborative effort, we established this platform that is a silencing RNA-based ferroptosis-inducing nanoparticles that we package with a silencing RNA for GPX4, which is a ferroptosis suppressor. And these nanoparticles are also coated with an aptamer that essentially confers specificity for CCA cells. When we test this platform, we see a reduction of tumor burden in our preclinical murine models. We also see an increase of ferroptosis markers, PTGS2 and ACSL4. And we see also reduction of tumor proliferation as shown by Ki67 staining in the tumors. Unfortunately, cancer cells learn to evade cell death, as we know, and over the past couple of years, we found that loss of function mutations of tumor suppressor and epigenetic metabolic regulator BAP1 seems to confer resistance to CCA, resistance to ferroptosis in cholangiocarcinoma. And then additional mechanistic studies shows that when BAP1 is lost, when there is a loss of function mutation for BAP1, there is an overall change in the cellular metabolism. One of them is a complete remodeling of the phospholipid composition, where we have an enrichment of ferroptosis blocking MUFA phospholipid versus at the expense of ferroptosis inducing PUFA phospholipids. And this was also essentially blocking specifically phospholipid peroxidation or damage of mitochondrial membrane, which is an hallmark of ferroptosis. We did a few more studies, and we identified this enzyme called TLCD1, which is an enzyme involved in phospholipid remodeling, as highly upregulated in BAP1 mutant CCA cells, and it seems to be also a stress-responsive genes in ferroptosis-induced cells. TLCD1 was also upregulated in two cohorts of CCA subjects from TCGA and our Mayo cohort. And additional cell-based assays shows that when we knock down TLCD1 in BAP1 deficient cells, we restore partially sensitivity to ferroptosis. So our current working model is that BAP1 loss upregulates TLCD1, which enriches for these ferroptosis-blocking phospholipids in cellular membrane, therefore blocking ferroptosis. And we hope to use our silencing RNA-based nanotherapeutics to block TLCD1 and potentially restore the sensitivity to ferroptosis in BAP1 mutant CCA. We have also additional goals to study a little bit the ferroptosis roles in the tumor microenvironment and identified additional metabolic vulnerabilities in BAP1 mutant CCA. This work could have not been possible without the generous support of the Stuart Matter Memorial Research Fellowship in memory of Stuart Matter, and we are extremely grateful for this generous support. And finally, would just like to acknowledge my team, mentors, and collaborators, and the Cholangiocarcinoma Foundation for their generous support over the past 2 years. Thank you.
So maybe I'll start with a question.
Yeah.
Okay, yeah, go ahead, Robert.
Thank you. Great presentation again.
Thank
so
you.
there are people with BAP1 germline mutations,
Yeah.
right? So do you see also resistance to ferroptosis, or is that specific to the cancer state, or potentially even cholangiocarcinoma?
Yeah, so we haven't specifically tested that. I know that in the germline, the mutation is heterozygous, in the tumor is homozygous, so you really need that for a tumor suppressor. And so the short answer is I don't know. It's a good question, and we probably have ways to check that. Yeah.
Okay, thank you, David. Yeah. And our final speaker is Haley Ellis from Mass General, speaking on resistance to mutant IDH inhibition.
Great. Well, I know I'm standing between you and dinner, so thank you for being here. I'm very excited to present a very brief overview of my fellowship research work. This is focusing on specifically one biomarker, IDH1, but I think it really represents the much bigger idea of understanding what tumors can teach us when treatment stops working. I really want to start by thanking Stephanie and Larry Dahl, who are sitting right there. And I have so enjoyed getting to know over the past year. They are just the kindest, most generous, incredible people. And this work would not be possible without them. Their excitement and passion for research and to push the field forward is so incredible. And here they are having a great time on the El-Bardeesy lab tour. I have really appreciated the encouragement from Stephanie over the months, and I've had very clear marching orders. So starting off with IDH1, which we've heard a lot about over the course of this meeting, this is an enzyme that's involved in energy production. And up to about 20% of intrahepatic cholangio, IDH1 is mutated, meaning there's a spelling mistake. And when that happens, this abnormal byproduct or toxic byproduct builds up in the cells. There's many downstream effects of that, one of which is actually helping the tumor become less visible to the immune system. We do have a targeted therapy approved for IDH1 mutant cholangiocarcinoma, ivosidenib or Tepsovo, which blocks mutant IDH1. And this does help. Most people get disease control, not tons of tumor shrinkage, but we know eventually the cancer progresses, and that's one of the big questions we had. So to study this, we used liquid biopsies. We analyzed circulating tumor DNA from patients who were treated on the phase 3 ivosidenib trial for at least 6 months. And we compared blood collected before treatment with blood collected when the cancer progressed. And blood gives us a really nice wide picture of what the tumor is doing across the whole body. And then we use this liquid biopsy results to identify some of these candidate outsmarting maneuvers, and then bring this to the lab and study how they potentially cause resistance. So the clearest pattern we found involved a group of genes in a pathway called MAPK. So in 5 of 18 patients, new MAPK pathway alterations emerged at the time of cancer progression. And these included mutations in genes like KRAS, which you've heard a lot about. Some patients had more than one of these mutations in their tumor, and some of them had very high levels. And really what was happening is that all of these different mutations were converging on this same pathway, MAPK, which is so important for cholangiocarcinoma growth. What we didn't see is also important. So what we know about IDH1 targeted therapy resistance is mostly from leukemia, where we also use these drugs. And most of the time there we see secondary IDH mutations, which in this cohort were very rare at low levels. So it certainly seems like what was the most dominant driver of causing this resistance was this MAP kinase pathway activation. And so this is where sort of the science really comes together. And prior work from our group showed that one of the important effects of ivosidenib, of Tibsovo, is restoring this immune response. And in our resistant models, this MAPK pathway activation didn't just give the cancer a new way to grow, but it actually helped the tumor hide from the immune system again. So it, blunted this immune signal that was restored by ivosidenib. What else could we learn from these liquid biopsies? We also looked at an even bigger cohort of patients on this trial at their blood samples at baseline and found that certain features predicted different outcomes. So patients that had ARID1A mutations or higher levels of mutant IDH1 in their blood seemed to derive potentially less durable benefit, whereas when we saw this mutation go away in the blood, people tended to do better. Now, this is not meant to exclude people from getting ivosidenib, but this is one of the first steps in really understanding these sort of markers of response and resistance so we can design better therapies. So where does this take us? Really understanding how resistance biology can allow us to get better care. How do we start smarter, treat smarter, and adapt faster? Using liquid biopsies as a tool to understand what is happening at the tumor at the time of starting. Combination therapies, you've heard a lot about RAS, which is really exciting. This is one of these important genes in the MAPK pathway that we commonly see in resistance. And so I think there's lots of opportunities to combine treatments as a way to hopefully delay or overcome resistance. And one of the common themes you've heard a lot about is liquid biopsies. How can we use these as tools to hopefully monitor tumor evolution over time and even be able to, hopefully in the future, our goal is to use these even before scans show any changes. So there's so many people to thank. I am just a small member of a team. Of course, I want to start by thanking the patients and families who participated on the Clarity Phase 3 trial. I want to thank Jinkai Wan, my lab mate in the Bardeesy lab, as well as the team from Servier. My clinical and research team at Mass General is just world class, and I'm so lucky to work with them. Getting to work with Dr. Bardeesy and Dr. Vijay and work from them and learn from them every day is such a privilege. And of course, the foundation, and again, Stephanie and Larry Dahl couldn't do this without you. So finally, this message from the foundation about strength and community and hope and research rings so true at this meeting. And I think when we're all here together in this room, patients, caregivers, advocates, clinicians, researchers, industry partners, we are all here for the same goal. So in honor of the stellar Stephanie, let's get back to work and kick cholangiocarcinoma's butt.
Yeah. Thanks, Haley. Do we have some questions for Haley?
You're also welcome to come talk to me at any point during the conference.
Sumera says everybody wants dinner. So
Yes,
anyway.
I imagine that too.
Yes. Well, thanks again. Thank you. Congratulations to all the awardees. Thanks for the great talk, and we look forward to seeing you at dinner.
So I'm just going to close the day today, thanking you all for staying with us right through to the end. I think it's been so exciting to see the new research through the posters, the oral presentations. What you've seen with the fellowships is not just good science. You are seeing our future leaders, and that's so inspiring. So I'm going to ask those future leaders, the young investigators, just remind them that there's a next-gen webinar tomorrow morning, 7 to 8 in room 250D, with breakfast starting at 6:30. For everybody else, general breakfast starts at 7 a.m. We're now going to move to the foyer to have a toast to the future before we go into the awards ceremony. And at the awards ceremony, we'll also be announcing the new fellows for 2026. And as you go past the archway, think about purchasing a globe if you're not yet done so. This is the pathway to the future to help fund our future work. Have a lovely evening. Thank you, everybody. Thank you.